I am pleased to have Dr. Maciej Czerwinski here with us today. Maciej received his Ph.D. from the University of Maryland at Baltimore Medical School in the Department of Pathology. He has been with XenoTech since 1999 and currently serves as the Director of Products Research and Development. Currently, Maciej directs the development of products and services for the analysis of drug metabolism and protein expression. Maciej also conducts research on the drug-drug interactions between biologics and small molecule drugs, and is the inventor of XenoTech's single-freeze hepatocyte product, CryoStax. It is my pleasure to present to you Dr. Maciej Czerwinski. Maciej?
Thank you, Matt. Good morning, everybody, or afternoon. Thank you for finding time to join us for this webinar on adverse drug reaction risks in genetically defined subpopulations. As Matt indicated, I'd like to encourage you to submit your questions in any form you'd like. We really thrive and are happy to see them. Adverse drug reactions are serious and common problems. Adverse drug reactions can reduce or eliminate therapeutic drug benefit, cause morbidity, and occasional mortality. Some adverse drug reactions may be due to patient genetic factors. Determinants of susceptibility to adverse drug reactions include pharmacokinetic factors, such as gene polymorphism in cytochrome P450 enzymes, other drug metabolizing enzymes and or transporters, and pharmacodynamic factors, such as polymorphism of drug targets, and vitamin K epoxide, reductase complex, VKORC1, and warfarin serve as an example of that type of situation. We think that adverse drug reactions that are based on known genetic variants are preventable. Both Food and Drug Administration and EMA issued pharmacogenomic guidance documents. The FDA has recommendations for early phase clinical studies, while EMA recommends in vitro studies prior to human exposure. These EMA recommendations involve, first, identification of enzymes catalyzing the in vitro metabolism, 2, characterization of metabolites formed through candidate major metabolic pathways, and 3, these two together, establishing the involvement of known functionally polymorphic enzymes in metabolism of a given drug. The third point addresses actually the heart of the problem, that is being aware of existence of high-risk subpopulation of patients and being able to define these populations in terms of non-genetic polymorphisms. Let's assume that a patient population receiving drug of broad therapeutic interest has varied abilities to metabolize it, as reflected by measures of exposure, either area under the curve or concentration at steady state. A narrow therapeutic drug would be represented in this type of graph by a line depicting on the left therapeutic effect and on the right depicting adverse effects moving closer to the center of the graph. Patients with high ability to metabolize the drug, let's say we call them ultra-metabolizers sometimes, and patients with a low or no ability to metabolize the drug have benefits of its therapeutic effect and do not experience adverse effects. But if a narrow therapeutic window drug is developed based on an average ability to metabolize it, ultra-rapid metabolizers, which are individuals in this area here, may not experience full therapeutic effect because this line would be moved to the right, to the center. Right? They may only receive a portion of therapeutic effect, while poor metabolizers may suffer for drug adverse effects when this line depicts closer to the center of the graph. This is a general scheme of mechanism of drug interactions that is based on enzyme induction or enzyme inhibition, or applies to those phenomena. But it also illustrates how genetic polymorphisms associated with drug metabolizing enzymes create subpopulations of patients with higher risk for adverse drug reactions. Genetic polymorphisms at the base of an increased adverse drug reaction risk are complex. By far, non-synonymous amino acid changes are most frequent, and Karolinska site for CYP alleles in humans lists about 400 examples of those, although effective single nucleotide polymorphisms can also be located in a 5' or 3' untranslated region of a given gene. Single nucleotide polymorphisms interfering with splicing can lead to no activity alleles such as CYP2C19 *2 or CYP2D6 *4. Same is for a single nucleotide polymorphisms. Decreasing allele activity such as CYP2B6 *6, CYP2D6 *41, or popular CYP3A5 *3. In the promoter region, extra TA pair in UGT1A1 allele *28 reduces gene transcription, while a single base pair change, C2G, in CYP2C19 allele *17 increases, have an opposite effect on gene expression. Gene copy number polymorphisms is also applicable to this phenomenon, and it's of particular interest in terms of CYP2D locus. Interestingly, this most complex CYP2D locus seemed not inducible, at least at the three best established nuclear receptor pathways. Drugs developed for treatment of diseases across all therapeutic areas are affected by major polymorphisms of drug metabolizing enzymes. Adverse drug effects include loss of therapeutic benefit, such as loss of a transplanted organ, recurrence of cancer, or lack of improvement in depression. Toxic effects include bleeding, thrombosis, or myelotoxicity. As you see from this tabulation, in addition to CYPs, UDP glucuronidase UGT1A1, and thiopurine methyltransferases are also implicated in a genetically determined adverse drug reactions. Polymorphism in drug metabolizing enzymes affects health by mechanisms other than metabolizing drugs as well. CYP1A2 and CYP1A1, both metabolizing caffeine, have effect on coffee consumption and therefore population's blood pressure. Other xenobiotic, nicotine, is metabolized by CYP2A6 enzyme. CYP2A6 poor metabolizers consume less cigarettes than CYP2A6 extensive metabolizer. They need less of them to achieve a pleasing level of nicotine in their brain, and therefore are at a lower risk for lung cancer. In many cases, adverse drug reactions are associated with subpopulations of poor metabolizers. CYP1A2, CYP2C9, 2C19, 2D6, and acetyltransferase 2, and others have genetically determined poor metabolizers. As you see from this table, prevalence of poor metabolizer varies among genetically and geographically diverse populations. Such genetic variation among different peoples suggests that the evolutionary advantage conveyed by drug metabolizing enzymes in interacting with environmental factors and they most likely would be nutritional. Please note that, for example, for N-acetyltransferase 2, there is really drastic difference between populations in Japan and in Egypt, between 10 and 90 spread. Also, I'd like to touch on CYP1A2, that this table indicates that the prevalence of poor metabolizers is about 12% in Caucasians. Alleles star 1C and star 7 are associated with a reduced activity of CYP1A2. They are both rare alleles. In our group of donors that we genotyped of about 180 individuals, we identify only one star 1C homozygote, and actually it was an Asian donor. In case you are interested in rare alleles from donors that are not represented in the few products that we currently have, the best way to inquire about existence or availability of such cells is to contact our account managers. It is observed that poor metabolizers are frequently associated with adverse drug interactions. These are examples of drugs implicated in adverse reactions metabolized by enzymes with variant poor metabolizer alleles. This table also illustrates a point that multiple polymorphically expressed enzymes may participate in biotransformation of a given xenobiotic. For example, fluoxetine metabolism is catalyzed predominantly by CYP2C9 and CYP2D6, with minor roles played by CYP2C19, 2C18, and 2E1. You may therefore consider that rare patients that are poor metabolizers for CYP2C19 and CYP2D6 and 2C9 may need a special safety consideration. For several years now, we have offered individual donor human liver microsomes characterized for variants in CYP2C9, 19, 2D6, 3A5, and UGTs 1A1 and 1A9. Microsomes for multiple donors with polymorphisms coding for high, moderate, or no enzyme activity are available in this product. Now we expanded our offer to include gene-known hepatocytes. Gene-known hepatocytes are pools of cells from donors with different polymorphisms having the same phenotypic effect. For example, CYP2D6 high activity pool comprises alleles star 1 and star 2 with varying gene copy number and other alleles. The advantages associated with using cells versus subcellular fractions include presence of transporters, physiologically relevant intracellular distribution of drug, and the presence of cytocolic enzymes absent from the microsomes. On the other hand, studies to identify major drug metabolizing enzymes involved in a biotransformation of xenobiotic are routinely executed with large pools of human liver microsomes and enzyme-specific inhibitors. These studies often have a disappearance of the test article as their analytical endpoint. Gene-known hepatocytes are formulated for confirmation of identity of a polymorphically expressed enzyme suspected of catalyzing formation of a given metabolite. Initially, such information may have been obtained from pooled microsome studies with specific enzyme inhibitors. When possible, cells from donors we identified are grouped into high, moderate, and no activity pools for this particular purpose. The gene-known cells can also be used to study metabolic pathways of a given drug in a genetically deficient population in contrast to normal population. For example, if a drug is predominantly metabolized by CYP2D6 in the majority of patients, it may be necessary to establish what secondary pathways of metabolism gave prominence in the absence of the major CYP, that is, in the CYP2D6 poor metabolizers. Characterization of gene-known hepatocytes include relevant polymorphisms in phase 1, phase 2 enzymes and transporters. Hepatocytes were characterized with pharmacogenomic array of 30 genes and about 160 single nucleotide polymorphisms. All assays that we use for a characterization are listed at our website. We supplemented the selection of assays presented by manufacturer of the array with few more relevant assays for CYP2D6, particularly including gene copy number, and others for CYP3A5 and UGT1A1. In relation to the complexity of CYP2D locus in humans, we are fortunate to have a support and collaboration with the lab of clinical pharmacology and Children's Mercy Hospital in Kansas City, Missouri. Polymorphism of CYP2D locus is particularly complex. In the array, we employ multiple normal, decreased, or non-activity alleles are included as listed in the table at the top of this slide. Activity score, on the other hand, AS, is the value that characterizes CYP2D6 metabolic activity without referencing any of the individual alleles present in a donor. It was devised to simplify translating genomic information into prediction of a patient phenotype. A donor with 2 normal activity alleles has a cumulative activity score of 2. Donor with 1 decreased activity allele and 1 no activity allele has cumulative activity score of 0.5. In our hepatocytes, as indicated by a box and whisker graph on the slide, there is an expected correlation between the activity score and dextrometorphan demethylase activity of the protein. The median enzyme activity declines in parallel to declining activity score, and the median enzyme activity is indicated by the vertical bar in the middle of the box. The bar and whisker graph also illustrates a significant variation in enzymatic activity in cells from donors with the same activity score. The variability determines the length of cumulative length of whiskers to the plus and to the minus side. You can also observe that decline in median enzyme activity is slight in comparison with a decline in activity score. For example, activity score 2 donors have median activity of the dextrometorphan demethylase only slightly higher than those located in activity score group 1. Donors comprising CYP2D6 high activity pool that we have activity score of 2.5, 3.0, and 3.5 respectively, therefore are individuals with a polymorphism in a gene. Currently available gene-known pools are CYP2C9 and CYP2C19, for which we have high and moderate activity groups. CYP2C19, 2D6, and 3A5, for which we have high, moderate, and no activity pools. Transporter OATP 1B1, for which we have a high and moderate activity pool, with allele OATP 1B1 star 5 being responsible for high activity. And for phase 2 UGT1A1, we have a high and moderate activity pools with stars with alleles star 27 and star 60 responsible for reduced activity of the enzyme. Future products include thiopurine methyltransferase and N-acetyltransferase 2 pools. The way we present characterization of genotype hepatocytes is illustrated in the case of CYP3A5, no activity pools. First, cryostack is a reference to pooled hepatocytes from multiple sources that were cryopreserved only once and have not been thawed before use. It is a differentiating feature, and I believe a beneficial one, of those cells over other products that rely on thawing cells and cryopreserving them for a second time after cells are pooled. Such cells arrive for a customer experiment, having behind them already 2 freeze-thaw cycles, which are detrimental to enzymatic activity of them. There are 3 donors comprising this pool. They all are homozygous for relatively frequent star 3, 3A5 allele that is associated with the reduced or no activity. Characterization of the donor is also given for polymorphisms of total 11 major CYPs. And please note that for most other CYPs, alleles present in these donors selected for these pools are of the wild type. Other example of characterization is that for CYP2C19, no activity pool. 3 individuals selected for that pool are all homozygous for STAR2 allele associated with no activity of the enzyme. Additionally, characterization of all the pools include metabolic activity of the enzymes characterized with 11 relevant marker substrates for major CYPs and demographic information combined for 3 donors present in each pool. I think this is all time that I have, and I would like to thank you for your attention, encourage your question, and we'll pass the microphone to my manager. Thank you.
Thank you, Maciej. As a reminder, if you have any questions regarding Maciej's presentation, please remember to type out your questions and send them in via the questions pane using your GoToWebinar user console. Before we move into Q&A, just a few quick announcements. As Maciej mentioned in his presentation, Secretary XenoTech currently sells 2 products, the first being the genotyped human liver microsomes. These are used to study the influence of allelic variants on the safety and efficacy of new compounds. These are supplied at 1/2 ml at 20 megs per ml of protein, and they're available as high, moderate, or no activities for CYP2C9, 2C19, 2D6, 3A5, UGT1A1, and UGT1A9. Regarding the Geneknown cryostacks hepatocytes, these are used to study genetic variants effect on drug metabolism and drug transport to identify risks of adverse interactions in genetically defined subpopulations, as Maciej mentioned. This is a pooled product, usually a pool of 3, but we can customize these for larger pools if needed, and these contain 4.5 million cells per vial. These are available in some degree with high, moderate, and no activities for CYP2C8, 2C9, 2C19, 2D6, 3A5, OATP1B1, and UGT1A1. One more thing to note, Secretary XenoTech recently launched a research biobank, and the idea behind this is we're really committed to furthering the knowledge surrounding hepatic diseases. And so what this is a collection of high-quality human tissue specimens representing early stages of alcoholic or non-alcoholic fatty liver disease. This tissue is collected in a timely manner with very precise care taken to minimize the downtime and preserve tissue viability. So we have 4 available products currently in our research biobank, consisting of normal donor liver prelysate, as well as donors diagnosed with steatohepatitis, steatosis, and we also have hepatocytes from select donors within our biobank with these diagnoses. There is a full donor list available online on our website, www.xenotech.com. And these tissues allow for the analysis of expression of drug targets and early markers of fatty liver disease. All samples come with a pathological diagnosis, demographic, BMI, history of diabetes, and alcohol use data. We also include multiple photomicrographs for each specimen, as well as H&E stain slides prepared for each lot to illustrate the overall tissue condition.